What's happening, guys? Arouge again, and today we're talking about Mendels Laws. He has 3 laws, and then we're going to finish it up with Punnett squares and how Punnett squares could be a helpful tool when determining patterns of inheritance. So, Gregor Mendel, remember, our monk from the 1850s, the father of modern genetics. He came up with 3 rules based on the experiments he did with his pea plants. The first rule, or the first law, we kind of already touched on, is the law of dominance. The law of dominance states, dominant alleles are expressed or shown, and recessive alleles are hidden if both are inherited. Okay, so remember, if we're talking about tall pea plants, if we get a genotype of heterozygous, of a dominant and recessive allele, the dominant allele, this dominant trait is shown, and the recessive allele is completely hidden. We can't see any trace of it, okay? The second law is the law of segregation, and these last two actually deal with gametes. They deal with how sex cells are formed. Mendel had no idea on meiosis, okay? Nobody even knew what a chromosome was, but he was able to deduce or to figure out that these patterns are true when sex cells are formed. So the law of segregation states that 2 alleles are segregated into individual sex cells. 2 alleles are segregated. What does segregate mean? To segregate means to split apart or to keep apart. 2 alleles are segregated into individual sex cells. We've already covered this too last week. Instead of alleles, replace that word with chromosomes. And we've already seen that. We know that our cells are diploid or diploid. And when sex cells are formed, those cells are haploid, and they only have 1 copy of every chromosome. Okay? So we still know this to be true. The last one, we're actually not going to cover this today. We're going to focus more on this at another day. But the law of independent assortment. The allele a gamete receives for one trait will not determine the allele for a different trait. So essentially, 2 different characteristics don't influence each other, okay? Today what we're going to focus on are these 2 right here, the law of dominance and the law of segregation. And we've already covered them, so today's going to be like a little bit of review and a little more in depth for those 2 laws. The law of independent assortment we're going to cover at a later time. So, the law of dominance, dominant alleles are expressed if and a recessive is hidden if both are inherited. We've already seen this. If we cross the purebred tall with a purebred small, what are we going to get? We're going to get 4 plants that are tall. This short trait is hidden. There's no trace of it, okay? We can't see a trace of that trait anywhere, phenotypically. Genotypically, we could see that it's here, but we don't know its genetics just by looking at it, okay? So that's Mendel's first law, the law of dominance. Dominant traits overpower recessive traits. Now, what about the law of segregation? So, let's say these 2 are hybrids. Okay, these 2 plants are hybrids. When we look at their offspring, we're going to get a 3-to-1 ratio. 3 talls and 1 short. Okay, so Mendel was able to deduce or to figure out that each plant has to have 2 copies of every trait. This has to have 2 copies, and this one has to have 2 copies. When they create offspring, each offspring is only inheriting 1 copy from each parent. So let's take a look at this. Let's make this connection to last week when we talked about haploid and diploid cells. Right now, both of these plants are diploid. They're 2N. They have 2 copies. I'm going to write that over here. They're 2N. They have 2 copies of every trait, but right now we're only focusing on plant height. Okay? When these plants form gametes, okay, those gametes are only going to be N. They're going to be haploid. They're only going to have 1 copy. Okay? So this plant is heterozygous, has 1 allele for being tall, 1 allele for being short. But when they formulate when they form gametes, those gametes are haploid and they're only going to have one copy of each trait. The destiny for these gametes is to fuse with another gamete, with another sex cell, okay? So how can we figure out the different options? Let me get rid of some of that red. How are we going to find the different options of offspring that these two heterozygous plants could have? Well, we use what's called a Punnett square. For the Punnett square, we put the alleles up top. Now remember, the alleles segregate. So they're going to be one here, and one here, one here, and one here. Okay? Those alleles represent the gametes. What goes into these four boxes represents the zygote. It represents the fertilized egg, okay, or the embryo. So in this first box, what options do we have? Well, we bring everything down and across. So this allele and this allele could join together here and give us an embryo that is homozygous dominant. This one right here, bring down this allele, bring across this one, this gives us an embryo that is heterozygous. If we go down here, this comes down, this comes across. We have another one that's heterozygous, and in this last box, we bring this down, we bring this across, and that gives us a homozygous recessive. Phenotypically, we have a 3 to 1 ratio of what Gregor Mendel saw. Okay, so if we go back to this slide, 3 plants were tall, 1 was short. If we go to that Punnett square, 3 plants are tall. This one right here is going to be short. Okay, let's do a little bit cleaner example with less mess, a little more organized. Tall plant that is homozygous dominant is crossed with a short plant that is homozygous recessive. Remember, this right here is representing a diploid cell. We need to represent the haploid cells, or the gametes, on top and on the side. Haploid, there should only be one letter on top and on the side of each box, okay? This represents a diploid cell. Segregate those alleles into haploid cells, and here we go. That goes down, that goes across. That gamete could pair with this one, okay? So let's backtrack real quick. What we're saying is this gamete could pair with this gamete. This gamete can pair with this gamete. This gamete can pair with this one. Or this gamete can pair with this one. That's really what we're saying, okay? If we find the genotypic ratio, how many are homozygous dominant, heterozygous, and homozygous recessive? We have 0 that are homozygous dominant, we have 4 that are heterozygous, and we have 0 that are homozygous recessive. Let's take a look at the phenotypic ratio. How many are going to be tall? 4. How many are going to be short? 0. Okay? Let's go ahead and do another one. A tall plant that's heterozygous is crossed with another heterozygous plant. We put the alleles up top and on the side. Again, this represents the possible gametes. This represents the possible gametes of the other parent. We have a homozygous dominant, heterozygous, heterozygous, and a homozygous recessive. The genotypic ratio, homozygous dominance, we have 1 right here. Heterozygous, we have 2. Homozygous recessive, we have 1. What about the phenotypic ratio? For talls, we're going to see 3. 1, 2, 3. Shorts, we're going to have 1. How about this one? A heterozygous crossed with a homozygous recessive. Pair the alleles. Okay, the genotypic ratio. How many are homozygous dominant? 0. How many are heterozygous? We have 1 and 2. How many are homozygous recessive? That would be the other 2. 1, 2. How many are tall? 1, 2. How many are short? 2. If you guys haven't noticed the trend, if we add all these numbers up, 0 plus 2 plus 2, these should always equal 4. If it equals any more or any less, there's a boo-boo, and you should double check it, okay? Okay. Now let's go into some word problems. Identify the genotype or phenotypes of individuals to find the allele combinations. So I'm not going to tell you what their allele combinations are. You have to read this on your own. For example, white flowers are recessive to purple. Cross a heterozygous flower with a white flower. Find the genotypic ratio, find the phenotypic ratio. Well, I'm not telling you what letters to use, but I am giving you guys helpful information. Right here, cross a heterozygous flower with a white flower. Okay, so what's dominant here? Well, if I go back here, white flowers are recessive to purple. That means purple is dominant. So I'm going to use capital P's. So to write the genotype for the heterozygous, I'm going to use a capital and a lowercase p. I'm going to cross that with a white flower. Well, what does that tell me? Well, I know that white flowers are recessive. The only genotype that gives me a white flower would be 2 recessive P's. Okay? Now that I have these alleles, now I'm in the business to find the ratios. Okay? Remember, these alleles are going to segregate. That would be the alleles from one parent. These are the alleles from the other parent. Now you do your Punnett square normally. Okay? Heterozygous, heterozygous, homozygous recessive, and down here would be homozygous recessive. Give you a 0 to 2 to 2 ratio, genotypically. Phenotypically, 2 are going to be purple, 2 are going to be white. Let's go ahead and do one more. Curly hair is dominant and straight hair is recessive. I'm going to use a capital C because that's the dominant trait. Okay? A woman with straight hair marries a man who is homozygous dominant for curly. Predict the genotypic and phenotypic outcomes for their children. All right, so what does this paragraph tell me? What information can I get from here? Well, I know that curly hair is dominant. Straight hair is recessive. The woman has straight hair. Well, if she has straight hair, I should be able to figure out her alleles because straight hair is recessive. Lowercase c, or a recessive C and a recessive C. The man is homozygous dominant. That tells me a dominant C and a dominant C. Now that I have the genotypes, segregate those alleles into the different gametes. Okay? Now I can fill in the Punnett square. What is the genotypic ratio? Well, there's 0 homozygous dominants. I have 4 heterozygous. And I have 0 homozygous recessives. What about the phenotypic ratio? All 4 of these children are going to have curly hair. 0 are going to have straight. Here's a couple more practice problems for you guys to do. Just basic word problems. Black hair is dominant to blonde hair. What is the genotype for an individual who has the alleles a dominant B and a recessive B? A dominant allele and a recessive allele. What do we call that genotype? What is the phenotype for an individual who has the alleles dominant and recessive? Okay, what am I going to see? And refer back to this information right here. What is the phenotype if someone is homozygous recessive for that trait? So what am I going to see if they are homozygous recessive? Okay? Go ahead and answer those questions. And last slide. Purple flowers are dominant to white. What is the genotype for a flower that has the allele combination of two recessive alleles? What do I call that genotype? What is the phenotype for a flower that has the genotype of two recessive alleles? Okay, what am I going to see? And what is the phenotype for a flower that is heterozygous for that trait? So what am I going to see if a flower is heterozygous, and use this information to help you, okay? That is it for today, guys, and I'll catch you all next time. Adios.